Okay, where were we? So, what I want to do now is talk more specifically about what lymphocytes are doing. And there are these two types of lymphocytes, the T cells and the B cells. In the book, this section is going through T cells, and the next section is going through B cells. But really, I want to kind of go a little bit back and forth between the two because there's a little bit of interaction, especially in how they come to be in the first place. But to begin with, something that applies to T cells that doesn't apply to B cells is that T cells are interacting with other cells. We often refer to T cells as being responsible for cellular immunity, whereas B cells are making antibodies, which is dealing with pathogens directly in bodily fluids. And so T cells have to interact with what are called antigen-presenting cells. And this is an example of antigen presentation. Now, the actual cell that this depicts would be a dendritic cell, which is not inappropriate, it's just not the type that we tend to really think about for this. So the antigens can be presented kind of in a couple of different ways. I'm actually not positive if both the ways necessarily are taking place within dendritic cells. And if they are, I doubt they're happening at the same time. But the picture kind of puts everything together. Now, antigens that are presented can be presented because they are foreign invaders that have gotten in and have been digested by phagocytic cells, and then the digested remnants of that are what are presented on the surface of the cell. And that's what is referred to in this picture as the antigen acquired from inside the cell, okay? Which is actually kind of a weird way to say it, but so here we have a pathogen, and it's been ingested by this cell. And I said earlier that when that happens in a phagocytic cell, the vesicle that contains the pathogen will merge with a lysosome, and that lysosome will release digestive enzymes, and it also has some acid associated with it. So it digests the pathogen down into smaller and smaller pieces. So these two little chunks here are supposed to represent pieces of the original pathogen that have been digested down. In this vesicle, after digestion's taken place, the antigens are going to be associated with what's called MHC. Now, MHC actually does stand for something, but for our purposes, we're not going to deal with what it stands for. We're just going to refer to it by its initials. What it does is more than what the original name means. So the name stuck, but we've learned that it does a lot of other things. So we just refer to it as MHC. And so it's represented by this thing right here, and it associates with the remnants of the antigen. So here we see the antigen attached to MHC within a vesicle that then merges with the cell membrane, and the MHC antigen complex is presented on the surface of the cell. So this is now an antigen presenting cell. Okay. So this is how we present antigens from pathogens that have been phagocytosed and broken down. The MHC that's used for this is specifically called MHC2, which hopefully immediately that makes you think, well, there must be an MHC1, and in fact there is. On the other side of this cell in this drawing, they're sort of showing a different type of antigen presentation. Now, I'm really not familiar enough with dendritic cells to exactly understand why they're showing it like this. Dendritic cells, like I said, are not the most common type of cell that we talk about here. But the antigen presentation that involves MHC1, which as far as the drawing is concerned doesn't look any different from MHC2, but it's labeled MHC1 here. The antigens that are presented here are antigens that are made in the body. And so down here it says acquired from inside the cell and acquired from outside the cell. That's kind of weird wording, but I think it's largely because I'm not that familiar with dendritic cells themselves. But in some other cell, and really all cells of your body do this, they make proteins on a regular basis. So whether it's fibroblasts making collagen, or red blood cells making the blood typing antigens, or neurons making neurotransmitter receptors, et cetera, et cetera. There's all sorts of different things that are being made. And each individual cell, when it's making those proteins for its own use, will take some fragments of those and present them on the surface with MHC1. Okay. Now, those antigens that are presented on the surface of cells that are things that are supposed to be, they're like collagen, neurotransmitter receptors, the blood type antigens, those things that I just mentioned, those are what we call self-antigens. There's things that are supposed to be in your body. This idea of antigen presentation, once your immune system sort of adapted to your environment, isn't terribly important, or we don't really worry ourselves about that because your immune system should not recognize self-antigens. And I'll explain how that comes to pass in a while. But why it's important is when a cell's infected, so if you get infected with the influenza virus and epithelial cells in your respiratory tract are being taken over by viruses and they start making new virus particles, the new virus particles that the cell's making while it's infected will be presented on the surface of the cell with MHC1. That is not a self-antigen. That's an antigen representing that something's gone wrong with the cell. Okay. So whether it's pieces of antigens that have been digested down and presented with MHC2, or antigens coming from some other source presented with MHC1, whichever it is, those are going to be antigen presenting cells. And I really, really doubt that this is going to happen simultaneously in one cell. Okay, I have no idea if this picture is just trying to put as much information into one picture as possible, or if it's trying to say that dendritic cells kind of do both things at the same time. And again, I don't know dendritic cells that well to say one way or the other. And it's not that important because, again, dendritic cells are the real big players here. Really what we want to think of is the presentation of pathogens that have been digested down by MHC2 occurs with macrophages and other phagocytic cells. And then presentation of antigens in regular everyday cells with MHC1, and by regular everyday cell, I mean your basic body cell. A lot of those will be self-antigens, which your immune system will ignore. But when there's presentation of a non-self antigen by a body cell with MHC1, that's basically a flag that that cell's been infected with something, or it's acting in some other diseased way. Cancer cells will often start transcribing genes that they wouldn't normally transcribe, and so those antigens will be presented on the surface. For instance, in colon cancer, one of the markers for colon cancer is a protein referred to as CEA, which means carcinoembryonic antigen. It's an embryonic antigen because it's something that's made in embryonic tissue. Normally, it should not be present in adult tissue because it stops being expressed after embryonic development. But in cancer, some cells start expressing that protein again, and so that's the carcino part of carcinoembryonic antigen. when that embryonic antigen is being expressed in an adult cell, that's pretty much proof that that cell is somewhere in the stages of developing cancer. So that would be a non-self antigen on the surface. Yeah. They just refer to as CEA. The E stands for embryonic because it's an embryonic protein. So this is antigen presentation. I want to talk about this to begin with because it's important in how T cells interact with antigens. B cells don't have anything to do with this because B cells don't interact with cellular components of disease. So this picture is out of the T cell section in the book, and it's explaining how T cells come to mature. Now, T cell really is just an abbreviation for thymocyte. A T lymphocyte is a T lymphocyte is a cell that's associated with thymus during this process. B cells are not associated with the thymus, so they don't fall into this group. But something related is happening in B cells also. Now, off to the side, there's little pictures indicating location of where each of these things take place. At the beginning, we have a picture of the femur and the thigh, and it's highlighting the bone marrow and the femur. The bone marrow is where these cells are made. So T cells and B cells, just like any other blood cell, is being made in the bone marrow. At that point, these are what the book calls untrained T cells, which they're very, very sort of, brand new cells. So they haven't started differentiating completely yet. That's indicated by in the parentheses, the two lines there just means that nothing's going on yet. The T cells then migrate from the bone marrow into the thymus, and that's why they're called T cells. B cells don't do that. Now, T cells are the ones that interact with antigen-presenting cells, so they have to be able to recognize MHC. So the first thing that happens for T cells in the cortex is they're subjected to a positive selection test, which is they're tested to see if they recognize MHC, that if they can actually interact with it, like they're going to need to do for the rest of their life cycle. If they do that properly, a positive result, they survive. If they do that inappropriately, if they don't interact with MHC the way they're supposed to, represented by this side of the picture, then they're destroyed. Okay. After the positive selection process, the T cell then moves on and it starts to express other proteins. Okay, so the completely untrained cell is undifferentiated, it's not expressing all of its proteins. But once it's gone through positive selection, it starts to become positive for these proteins that are referred to as CD4 and CD8. And those are actually going to be part of interacting with MHC later on when they're dealing with actual antigen-presenting cells. At this point, they then move on to another test, which is still taking place in the thymus, which is referred to as negative selection. So in positive selection, they survive if they do what the test is asking them to do, which is to react with MHC. In negative selection, here they're exposed to antigens, and the T cell receptor is tested to see if it reacts with the antigen. The antigen that it's being exposed to, or the antigens that they're being exposed to, are self-antigens. And this is part of that early developmental process, that your adaptive immune system has to adapt to your environment. And so if the T cell receptor does not bind to the self-antigen, then that tells us that it is not going to be the result or be the cause of an autoimmune reaction sometime later in life. So we keep those because it's not going to react with anything in our body. If it happens to react with one of those self-antigens, it's destroyed. Okay? So this is negative selection. The positive result, if it binds to the self-antigen, it dies. The negative result, if it doesn't bind to it, then it survives. Okay? Then we have cells that have gone through this process. They finally differentiate into either CD4 positive or CD8 positive cells. Okay? And they leave the thymus and move on to the secondary lymphatic structures. The picture there suggests a lymph node, which is sort of the common example, but it might move on to the spleen or to a tonsil or lymphoid tissue somewhere in the digestive tract or whatever. Moves on into a secondary lymphoid structure. Once it's there, it's a mature cell, but now it needs to be activated. It's just waiting to see if it will interact with the antigen that it's specifically for. Okay? There's no telling whether it will ever interact with that antigen. Okay? It's a matter of whether you're ever going to be exposed to it. Now, ideally, you're kind of making an infinite number of lymphocytes that are making each a unique T cell receptor or antibody for B cells, so that you have the ability to interact with an infinite number of antigens. But that's not true. You can't make an infinite number of cells. So you make a huge number of cells, but there is an upper limit to how many can be made. So you're not going to be necessarily immune to everything out there, but the numbers are there to kind of improve your chances of reacting to most things. What's called a mature T cell here is then going to move out into, well, sorry, I just said that. The mature T cell is out in the secondary lymphoid structures. And there it can interact with the antigens that are actually getting into your body and potentially causing problems. So this picture is showing us the CD8 cell, which is also called a cytotoxic T cell. So the subscript C there refers to cytotoxic. So it's a mature cell in the previous picture. It's called naive here because it's gone through the testing, the positive and negative selection, so it's mature. But it's not yet activated. It's naive because it's never interacted with its particular antigen before. Now, what the naive cell is going to do is eventually be presented with an antigen by an antigen presenting cell using MHC1. CD8 and MHC1 interact. Okay? CD4, which we're going to see in a second, won't do the same thing. So cytotoxic T cells only interact with antigen presenting cells using MHC1. And earlier I said that MHC1 is presented on the surface of your average everyday body cell. At this point, if we get a naive T cell that's gone through positive and negative selection, if it's reacting to an antigen, it's not reacting to a self-antigen. We've gotten rid of the lymphocytes that do that. So instead, if it's reacting to an antigen, it's reacting to the antigen presented by a cell that's been infected or by a cell that's initiated some other disease state like cancer or something like that. Okay. In the picture here, they're using a dendritic cell, and again, dendritic cells are not my area of expertise, so I don't want to say that this is wrong, but it's a little easier to explain this if we talk about a diseased body cell presenting here. So if you've been infected with the influenza virus and the virus has gotten into cells in your respiratory tract, some of those cells will detach from the respiratory tract and move into the lymphatic system. And then they'll end up in a secondary lymphoid structure where this will happen. and this T cell will then be alerted to the presence of infected cells. Okay. Once that happens, once they interact here, CD8 and MHC1 and the antigen and T cell receptor all play a part, this cell ends up getting activated. Now, the book actually leaves out in the picture what actually causes the activation. When all of this occurs in the picture, additionally, a chemical is released. It's a cytokine, is released to tell the T cell to become activated. When it gets that signal, it then starts dividing to make multiple copies of itself, what we call clones of it. Okay? That signal might be coming from the antigen presenting cell, or it actually might come from another source that I'll show you in a second. But whatever that source is, the chemical here tells it to be activated. It starts making copies of itself. And it differentiates into two different types of T cells. There's the activated T cell, which is going to go out and destroy other infected body cells. And then there's the memory T cell, which won't do anything right now, but if there's a subsequent infection of the same pathogen in the future, these cells are ready to fight it off, and they don't have to go through this activation process again. Now, in the book, this is the next picture they show, and I went to this because more stuff's labeled here, but really, I would have preferred a little bit to start here with this picture, which is kind of the exact same thing I just showed you, except the labels are different. This is a CD4 positive cell, which is called a helper T cell, and it's interacting with an antigen presented with MHC2. Okay. So CD4 and MHC2 have to go together, and the T cell receptor and the antigen come together, and this cell gets activated. Okay. Now, the cell that's presenting the antigen to the helper T cell, or the CD4 positive cell, is going to be some sort of phagocytic cell. And so it's going to release a co-stimulant to tell the helper T cell to then make clonal copies of itself, including memory and active helper T cells. This picture is supposed to represent what a helper T cell does. This is, I guess, a vesicle of some sort inside the cell, and this is a vesicle that's releasing its content into the surrounding tissue. And those are cytokines. So going back to this picture here, which it would get kind of overly complicated to actually put all of this in, but if the antigen presenting cell, infected body cell or something like that, can't release a signal to help activate the cytotoxic T cell, sometimes the body cell is just so far gone it's not able to carry out all of its normal reactions. So if the chemical signal isn't released here, helper T cells will release the signal and help the other cells become activated. That's why they're called helper T cells. Now, the picture in the book that really shows the helper T cell the best is a helper T cell helping a B cell become activated, okay? Which, it's kind of odd. This is sort of way at the end of the B cell part of the chapter, but it really kind of fits in with the story here. So here's the helper T cell that was activated previously, and it releases cytokines that help the B cell become activated, okay? Now, it's nice to have this picture showing the helper T cell interacting with the B cell, but this picture actually isn't showing how the B cell gets activated. So I have this other picture here, and this is again from the B cell part of the chapter, which is very similar to the activation process that was described for the T cells. It's presented slightly differently though. Here we have 3 different B cells, and on their surface they're using antibodies as receptors. So usually we think of antibodies as being dissolved into the plasma or other fluids. But actually, for the activation process, they're on the surface of B cells. In this picture, they have the blue-colored ones, the red-colored ones, and the purple-colored ones. to represent 3 different antibodies. Okay, so these are 3 different lymphocytes. They each make a unique antibody, and so they've color-coded them so that you can say, oh, they're different ones. The antigen is represented by this triangle, and we see an antigen binding to the antibody on this B cell, to the red antibodies. It's not binding to the blue or the purple antibodies. Okay, so it's specific for that antibody, not the other ones. When it binds to the antibody here, and this is a naive B cell, first time it binds to the receptor, it binds to the antigen, it then forms clones of itself. So here we have a couple more with red antibodies on them. And calling them clones is important because clone means the DNA is identical. Okay? Clone does not mean Arnold Schwarzenegger in a movie. Okay? In movies, when they talk about clones being like carbon copies of people, that's not exactly what clone really means. Clone just means identical DNA. Okay? And so having identical DNA, this lymphocyte and this lymphocyte are going to make the exact same antibody as this lymphocyte, which is color-coded red. Because it has that variable region that's unique to that lymphocyte, all of the clones of that lymphocyte are going to make the exact same antibody, which is against this antigen. The clones are going to start dividing and making more and more of themselves, and they're going to be memory B cells, and then the active B cells are called plasma cells. They're called plasma cells, and they look different because all of this squiggly stuff in the cell is supposed to represent the rough endoplasmic reticulum and the Golgi apparatus, which are organelles important for secreting proteins. And so the plasma cells secrete a bunch of copies of the antibodies. Okay? And then those antibodies are available to bind to whatever antigen they're specific for and help the immune system get rid of them. Now, in this picture, they expand a little bit more. This is the activation process that happens when a naive B cell is first exposed to its antigen. Memory B cells have already been activated because they've been exposed to that antigen for the first time. If the antigen shows up again in a subsequent infection, the memory cells recognize that antigen and immediately start dividing and making more memory cells and more plasma cells, which start secreting the antibodies. Okay? Up at the top, these 2 events are described as the primary response and the secondary response. The primary response, the first time you're exposed to a pathogen, this type of stuff has to happen. Not just with the B cells, it's happening with the T cells too. But then when the memory cells are made at the end of that activation process, the next time and all subsequent times you're exposed to the antigen or the pathogen, you're going to have this secondary response, which is much quicker. Just the memory cell starts dividing and making a bunch of new cells ready to fight off the infection. It doesn't have to go through all of the steps here, so it happens much quicker. It's in the picture, so I want to point that out here, but we're actually going to come back to that idea a little bit later on to talk about that. Okay. So across these pictures, from this picture here with the T cell to this picture here activating the B cells, this is sort of the life cycle of the lymphocyte. It's born in the bone marrow. The T cells go to the thymus. The B cells stay in the bone marrow. And T cells have to go through positive selection to be sure they recognize MHC. And then both of them go through negative selection to be sure that they don't recognize self-antigens. And then the mature yet naive lymphocytes move to secondary lymphoid structures where when they first encounter their actual antigen, they're activated. They make clonal copies of themselves, which differentiate into the active form of the cell and memory cells. And the memory cells are there to persist for future exposure to the same pathogen. That's sort of the life cycle of what's happening there. Any questions about the processes that I've just described?
Just backtracking a little bit, just when it comes to the initial antigen presentation with MHC2, that is that just corresponds to antigens that have been partially digested,
Right.
versus MHC1, which is another source, an antigen from another source that has been not digested. Right.
Not exactly. In the book, they're using a dendritic cell, which is a somewhat unusual cell. For MHC2, think of those with phagocytic cells. And what was described there was phagocytosis. But think of, actually, let me go back. So think of this picture here of the inflammatory response. Bacteria have gotten in through this wound, and they're being phagocytosed by this neutrophil. Okay, they are digested within the neutrophil, and then the pieces of them are presented on the surface of the phagocytic cell to the helper T cells. Okay, so the phagocytic cells use MHC2. Helper T cells have CD4. Those two correspond. Okay. If any of the bacteria get past the phagocytes and they cause an infection in one of these cells, or if it's a virus doing the same thing, some of those infected cells will present antigens, which are antigens that are signs of infection. Okay. So I think the way you said it a second ago was the digested bits associated with MHC2, which is correct. And then I think you said undigested or something like that?
Or otherwise not phagocytized.
Right. So that's a little bit of a confusing area to get into, because really it's more that, yes, they're not phagocytosed because it's not in phagocytes that it happens. It's in other body cells that are making antigens. So,
Like the cancer,
right,
CDAs,
right, that kind of stuff. So just within vesicles that come from the protein synthesis pathway, some of those will get MHC1 associated with the antigen, so that's presented on the surface. And if it's a normal cell that's not infected, it's presenting self-antigens, which is okay. If it's an infected or diseased cell of some sort, it's going to be presenting unusual antigens because it's doing something strange, whether it's making new virus particles or it's cancerous, it's making unusual proteins, et cetera, et cetera. There's a bunch of different reasons why cells can present weird antigens with MHC1. But MHC1 interacts with CD8. So that's what's presented to the cytotoxic T cells. Okay. And then the cytotoxic T cells, after they've been activated, they go out into the various tissues of our body looking for more infected cells that are presenting those antigens also. And so the same thing happens. You've got the cytotoxic T cell with CD8 interacting with infected body cells with MHC1. But now, instead of them just being activated, when they're out doing their job, when they find an infected body cell, they destroy it. Okay, and they use some chemicals called Granzymes or perforins to do that. Now, Granzymes and perforins are also used by natural killer cells, but natural killer cells are not doing it against a specific thing based on an antigen. They're doing it on things that are not that are being recognized by the pattern recognition receptor, or whatever that was called earlier on.
Yeah, I mean, in these two processes, that's a concerted action. It's not necessarily that one occurs before the other in terms of cytotoxic versus T cells.
Actually, a little bit better explanation of that is the cytotoxic T cells that are going out to do this probably are too few in number to really get all of the infected body cells. So they'll recruit the natural killer cells to help out and tell the natural killer cells which cells to get and destroy. Okay. So it's not one before the other so much as they work together. Yeah. So talking about cytotoxic T cells and destroying things, I want to go to another video. So I have a few other videos about the immune system here. Oh, shoot. But I got to go to a different. Come on. I actually have this video in a different class section. So go back down here, and there's the one we watched a little bit ago. And then there's some other immune system videos. Whoa, where is it? Oh, I'm sorry. I didn't scroll down far enough. Maybe it wasn't yours, and I just didn't get there. But this says killer T cell attacking cancer. And killer T cell is another name for cytotoxic T cell because they kill things. This video is on YouTube, and it's publicly available on YouTube. To embed a video from YouTube into Blackboard, it has to be a publicly available one. I actually want to show you a different one, which is on this website that I have a link to. The one I'm going to show you is actually by the exact same people that made this one. It's just a more recent video with better data. And it's a little longer with more information. So let me go to this page here. And this is just going to show you cytotoxic T cells doing their job. Now, the video I watched earlier with the phagocyte chasing down the bacterium, we're going to see something very similar happening here because the T cells move in the same way. And this video is really pretty incredible because it's some of the best cellular video that I've seen representing this. So let me just play this out. It's got a voiceover explaining what's happening. There's a lot of credits at the beginning here. I have no idea what that is. And this is between some Cambridge and some NIH researchers collaborating together. Cue the music.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body. They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer cell, an explosion of membrane protrusions explode the surface of the target. Cytotoxic T cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarized from the rear of the T cell to the blue cancer cell. These toxic granules secrete specifically toward the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell. Movies like these allow us to study the efficiency with which cytotoxic T cells operate. Now we'll leave you with a few more movies of T cells doing what they do best, killing cancer.
Okay. So the rest of it is more credits. Now, I really like at the beginning here where they're showing this one phagocyte, and we're actually following it along. What they're doing here is they've used some sort of fluorescent marker, which might be a protein, a membrane marker or something like this in this example. And then this is in some sort of, I think, tissue based on the way that it's moving around. So there are other cells that it's moving through. We just don't see them because they don't have the same marker as this. I really kind of wish they'd had like a couple of frames at the beginning with regular light so you could see the cell landscape that this is all taking place in. But there's that. Now, you probably can't quite see up on the board, but right here, there's another cell, and over here there's another cell that are very faintly lit up. And we saw in a couple of other places, shoot, that's not it. I don't think I can catch the exact frame, but there were a couple of others that it moved past. Yeah, like this. So there's more than just that one cell. There are a few others. So those are the T cells that are marked, and we're just seeing it move around in there. And then they switch over in the color scheme. So now we have the T cell in this kind of yellow, orangey kind of color. And then in blue is a cancerous cell. And they're showing how it's interacting. This picture here might as well be, oops, this picture right here, or this picture right here. Okay. For the sake of a textbook, they're always going to highlight the molecular structures on the surface like this, making them much larger in scale than they really are. But really, that's happening where the two cells are coming together. So MHC and CD8 and MHC, CD8, and the T cell receptors that are playing a part in all this are inside where these two are coming together. Okay, and then they switch over to where they start showing, oh, not quite far enough, T cells that are latching onto these. But when they release their contents, the Granzymes and the perforins, the cell suddenly flashes bright red, indicating the presence of those I want to catch a frame where it's actually lit up bright red. There. So that bright red color is just indicating that there's a chemical change inside the cell because of what's being released by the T cells onto it. That's happening inside. In these pictures, the Granzymes and the perforins look like they're diffusing across the space between the two cells, which would mean that they could potentially interact with other cells, and it'd be more like a shotgun blast to the area rather than a very directed injection into that cell. So I like this, makes a very good case for what it really looks like. And then they just run through a number of examples of that happening that they measured. Okay. So like I said, I just found out about this, I think it was last week when my colleagues sent the link to me. And it was only posted at the beginning of February to begin with. But it's, I think it's a really good movie to show you what cytotoxic T cells are really doing. What's shown in a book like this is always necessarily a little bit oversimplified, but the idea is that it's sort of destroying those cells by injecting them with these Granzymes and perforins. Now, Granzymes are enzymes found in the granules that they mentioned in the movie. That's where they get their name. What they do is they digest macromolecules inside the target cell. So it basically, I guess you could say, liquefies the cell from the inside out, which isn't a very accurate description, but it's just digesting macromolecules inside the cell. What perforins do is they perforate the cell membrane. So it's kind of like the membrane attack complex for complement. Only membrane attack complexes are getting after the bacterial pathogens themselves. Perforin is used to destroy the infected body cells that are present. Okay. So I like that movie. It does a good job of showing you what's happening there. Now, the next thing I want to talk about, so the video I was just showing you there was basically what T cells do. And so the next thing I want to get into is what do B cells do? And essentially what B cells do is they secrete antibodies. But there's a bit more to be said about antibodies. And so this table here, which is coming after the introduction of what antibodies are in the B cell section, is showing us the different types of antibodies. Now, a single lymphocyte, or then all its clonal copies that are made after activation, make the same antibody in the sense of the antigen binding site at the end. Okay, so that where the Y part of the thing is reaching out, sort of like where my hands are, that's unique to that particular lymphocyte, and all the antibodies it makes have the exact same thing there. But it can actually make 5 different types of antibodies, which is really more about what's happening at the other end, not the part that interacts with the antigen, but how the antibody interacts with other things. Okay. Now, the most common type of, before I get there, immunoglobulin is just another word for antibody. It's abbreviated capital I, lowercase g. So when we're talking about these, they're referred to as IgG or IgM or IgA or E or D. So the most common of them, making up about 80% of the antibodies in your system, are IgGs. In the picture of, almost there, the plasma cells that are just cranking out antibodies here, these would be IgGs primarily. Okay, that's basically what's being released there. They're found in monomer forms, meaning just one antibody, and they're secreted into the plasma, where they can then stay within the plasma or move, if needed, into other tissues where blood vessel permeability has been reduced by the inflammatory response. They're really good for neutralizing antigens like toxins, so they can't be toxic. And by themselves, they're going to opsonize the target. Okay, opsonization means that it enhances phagocytosis. Yeah.
What do you mean they stay in monomeric form, or is that implying they stay in tertiary structure?
It's that they stay in this sort of Y-shaped
Well, you
thing.
just know with like different levels of protein. Like
No, it's not about protein structure.
well,
They
sometimes-
have quaternary structure.
I just got confused because sometimes monomeric proteins are associated with tertiary structure before they go to quaternary structure.
No, it's not talking about that.
Polymer.
These have multiple subunits, the light chains and the heavy chains. So they have quaternary structure. When I say monomer, I mean a single antibody, okay? Which in the other ones I'm going to talk about, we're going to see other types of structures, okay? So opsonization would mean that the antibody binds to the surface of a pathogen, and it makes it easier for phagocytes to do their job and ingest them. That's what opsonization means. Okay. The next most common, according to this table here, at 30%, are the IgAs. And these are found primarily in dimer form, meaning 2 antibodies connected together. Okay. Now, they can be in a monomer form under certain circumstances, but generally we see them in dimers. And they have an additional component, which is called the secretory component, because they're primarily secreted into various exocrine secretions. So they have mucus, which would be what's secreted across any mucous membrane, tears secreted by the lacrimal glands, saliva from the salivary glands, and then colostrum is referring to mother's milk. That's what's secreted from mammary glands. For everything but the mother's milk, primarily the point there is that they're present on the surface of these barriers, whether it's the skin or the mucous membranes or the conjunctiva over the eye. They're present there to bind to antigens right away. So if those antigens ever get into the body, they're already attached to an antibody, and so white blood cells are ready to go ahead and get rid of them. Okay. Now, the mother's milk part of it is important because that means that they're passed on to the newborn. The adaptive immune response is not ready to go right when somebody's first born. So to help a newborn be immune to things, it receives passive immunity directly from the mother. Some of that comes through the mother's milk, which is mentioned here, but actually the majority of it comes in the form of IgGs that are passed across the placental barrier. Okay. And really what it is a woman is immune to various things that she's been exposed to throughout her life, and she passes that on to her fetus because likely the newborn is going to be existing in the exact same environment. So it'll just sort of inherit that immunity directly through the placenta. The mother's milk situation is rarely that necessary, but if a woman has given birth to a newborn and then is exposed to a pathogen and she develops an immunity to it, then she can pass that immunity on to the newborn before the newborn's able to develop its own immunity to something. Okay? So that's not going to happen too often. Most often, there's not going to be some new pathogen that shows up right after the baby's born. But if it does happen, then that immunity, when the mother gets it, can be passed on. Yeah.
Are IgAs also present in, once the colostrum matures into mature maternal milk, I guess you could say? Are IgAs present in that as well?
I would think probably so, but this is the first source that I've actually seen referring just to colostrum and not just mother's milk in general. So I think probably it is always available through that, but because it really does depend on, if the mother's exposed to something after she's given birth, she has to develop her own immunity and then she wants to pass it on. So it's only very initially, then it's not going to be very helpful later until the newborn can develop its own immunity. So, like I said, most sources won't necessarily just say colostrum, but we'll say just mother's milk in general. The next most common type, according to this, are the IgMs at about 6%. What makes IgMs sort of unique is that they form pentamer structures. So 5 separate antibodies will all be linked together, forming this major super antibody that has 10 antigen binding sites on it. And IgMs are great for binding to a bunch of copies of an antigen. So when you're infected with something, you don't just get one virus or one bacterium. You get a whole population of bacteria. You get a whole, I don't know what you would call a bunch of virus particles. A buttload of virus particles, whatever. So you want to bind up as many as possible. And IgMs are great for doing that. So it can bind to 10 different things, and then another IgM can bind to one of those and 9 more of them. And then you have this big clump of 19 things, and that can keep happening. You just get this huge group of things all together. When that happens with cells, we call that agglutination. Okay? And hopefully you recognize that word because it came up when we were talking about blood typing. when you're doing blood typing testing, you want to agglutinate blood cells, or well, you want to see agglutination somewhere. If somebody has the A antigen on their red blood cells and you test those red blood cells with a serum that contains the antibody against the A antigen, then you'll see agglutination of red blood cells, which is a positive result, and that person has type A blood. Okay. Or the same thing for type B or for the Rh factor for positive blood types. If you don't see agglutination, then the person isn't that blood type. So I guess if you want to say you want to see no reaction because you want O negative people, and you got more O negative blood to for the Red Cross to get. That's right. But that's agglutination, which is really a big thing that IgM does. Okay. The next most numerous one, which is barely numerous at all, at about 1% according to this table, are the IgDs. And they are B cell receptors. Okay. So as we're looking at these these B cells, the memory B cells here, the naive B cells here, they have antibodies on their surface acting as receptors. Those are IgDs. Okay. Now, there's a weird little thing in the book here, which I'm not an expert on and I haven't researched to really look into too deeply. But right here it says IgD B cell receptor. Down here under IgM, it says the monomer form of IgM serves as the B cell receptor, which is not internally consistent for this table right here. It says this is B cell receptor and this is B cell receptor. I've always known the IgD is the B cell receptor. I've never heard IgM being that case. It might be, and I have no idea if this is true, but it might be IgDs are the B cell receptors on naive B cells, and IgMs are B cell receptors on memory B cells. I might be making that up, but that would explain what seems to be a contradiction there. Okay. And the book doesn't go into that, or the table doesn't go into that. And then the least most common one here at 0.002% prevalence are IgEs. And IgEs are known for associating with mast cells and basophils. Okay. Now, mast cells are the cells that release histamine that initiate the inflammatory response. So if there's an IgE in the surface of a mast cell acting like a receptor there, if an antigen binds to it, that's going to cause the mast cell to be activated and release its histamines. That's essentially what is happening in an allergy. Okay. If the antigen that's binding to the antibody is not a pathogen, but causes mast cells to release histamines, then we would refer to that as an allergen, because it's binding to that and causing the mast cell to create inflammatory response without having to actually fight off an infection. And so, depending on exactly where that's happening, you might be producing excess mucus in your nasal cavities. You might produce hives somewhere else, or it might cause blood vessels to dilate and tissue perfuse. And you get angioedema, which is the swelling that you see in anaphylactic shock or anaphylaxis, which I think has been best depicted in the movie Hitch, starring what's his face, Fresh Prince of Bel-Air, Will Smith. Yeah, there's a great sequence in there where he eats something and he blows up and his whole face gets scary. So it's that kind of reaction there. Now, basophils are the least most common type of white blood cell. And for the most part, we tend to ignore them in this class because they're so rare. But they seem to have some relationship to anti-parasitic activity, which is what it says there at the bottom. And some sources will actually go so far as to say specifically parasitic worms. So that suggests that there's something about IgEs that will help to activate basophils, which can then help fight off an infection of parasitic worms. Now, the the podcast that we listened to about Jasper Lawrence, he was talking about a relationship between the parasitic worm, the hookworm, and his allergies. In that podcast, it didn't say anything about the basis of it at the molecular level, but it is kind of curious in light of what we heard in that podcast that IgEs are associated with the two cell types that are probably related to what's going on there. I have no idea what mechanism might be at the root of that, but maybe part of what is going on with Jasper Lawrence's story is that with the parasitic infection, IgEs are preferentially going to be associated with basophils, help fight off the parasitic worm, and therefore mast cells are not going to be picking up too many IgEs, and therefore people are less likely to have allergies. Now, that's not at all the mechanism they were talking about. They were talking about these lullaby cells, which is another type of lymphocyte that can play a role in there. And I'll point that out in a little bit. But it is kind of curious that the two things that are described in happening in Jasper's story are associated with the two cells that are related to this very, very rare form of antibody, the IgE. So I don't know what to make of that, but it always caught me as interesting to see. Now, in the book, which is where this table is coming from, they're talking about what these different types of antibodies can do and function-wise down here. I'd like to present a slightly different way of thinking about that, which will hopefully make it a little bit clearer. So when antibodies and antigens form a complex, several things can potentially happen, which were basically explained in the function part of the bottom of that table, but I want to lay it out a little bit differently. To remember what's happening, I like to use a mnemonic, which is Plano. Okay. Now, when I first started doing this, one of my early sources in teaching presented it this way, but actually used the mnemonic of plan. I added the O on the end because I think it's important to add that into this. But each of the letters here stand for one of the things that can happen when antibody-antigen complex forms. P stands for precipitation. Precipitation is when something falls out of solution. I should have thought to look this up earlier. I was talking about this with my morning class, and I was trying to describe this particular precipitation reaction, and I can't remember the exact players, but you can take two salts, and by salt I mean an ionic compound made up of components on either side of the periodic table. So sodium chloride is a salt because sodium is in the first column and chloride's in the second to last column, and they make an ionic bond. So you can take two salts like that, maybe sodium chloride and I can't remember what I don't actually remember if it's sodium chloride. Now, I got to look this up. It's bothering me. Oh, I am right. Okay. Oh, why can't you be in an animation? Okay. So sodium chloride and silver nitrate, when they're combined, the silver gives up the nitrate and picks up the chloride. Sodium gives up the chloride and picks up the nitrate. It's not exactly how it works, but basically. And so there's a substitution reaction takes place where you get silver chloride and sodium nitrate. I always thought sodium nitrate was explosive. I must be wrong. But Oh, here's the animation. Yeah, okay. So here's a beaker with a solution in it. And it's probably a solution of silver nitrate. And the dropper, they're going to add sodium chloride solution to that. So let's see what happens. Oh, come on. There we go. So you're taking two clear solutions and adding them together. And when they mix, what happens is this cloudy stuff shows up. If they ran this animation a little bit longer, the cloudy stuff would settle at the bottom. And that's what's called the precipitate. Okay. So that's what precipitation means. In the sense of antibodies and antigens, what this means is small antigens will fall out of solution. Okay? And by a small antigen, I usually mean a protein antigen. So if you get some sort of toxic protein into your system, antibodies bind to that. They make this big complex of antibodies and antigens. And it's too heavy of a complex to stay suspended in the solution of plasma. So it falls out of solution. Okay. That's what precipitation is referring to. L stands for lysis. And lysis means to break apart or to destroy. Really, it doesn't fit with the mnemonic, but really we should call this cytolysis. So the cyto in cytolysis means cell. So cytolysis means destroying a cell. Okay. And that is what happens in complement. Okay. So this is the result of the classical complement pathway. So that picture I showed you before, antibodies bound to the pathogen, C1, complement protein 1, interacts with the antibodies. It recruits C2 and C4, which then come together in partially to make the C3 convertase, which changes C3 or breaks up C3 apart so that C3b can do the same thing to C5, and then C5a recruits C6, 7, 8, 9 to make the pentamer attack complex and lyse the cell. Okay, so the classical complement pathway is lysis. A stands for agglutination, which I just talked about. Okay, that's clumping. That's when large pathogens, antigens, which usually means cells, clump together because they're connected to a bunch of antibodies. Agglutination is often associated with IgMs because they're in these pentameric forms. Okay? Precipitation and agglutination are kind of the same idea. It's just precipitation is when a small antigen binds to antibodies, and agglutination is when a large one does. Okay? But basically, they're both kind of falling out of solution to some degree. And then N stands for neutralization, which is when the antigen or pathogen is inactivated. So if the antigen's a toxic molecule, if it gets covered in antibodies, then it can no longer do whatever it does as a toxin, maybe. If the antigen's a pathogen, like a bacterium or a virus, if it's covered, it won't be able to do its job either. With some bacteria, if they have some sort of motile structure, like a flagellum, if the antibody binds to the flagellum and makes it unable to move anymore, that neutralizes or sort of freezes the pathogen in place. But for like a virus, if you just coat the virus in antibodies, it won't be able to interact with its host cell. It'll be neutralized there. Those various things there. So really those four are the traditional examples of what happens between antibody-antigen complexes. At the end of any of these, really what happens to the precipitated antigen or the lysed antigen or the agglutinated antigen or the neutralized antigen is that it's then phagocytosed to clear it out of your system. But sometimes the phagocytosis is all that happens. And that's what the O stands for. That's opsonization. Opsonization means enhanced phagocytosis. So antibodies will just mark an antigen as being foreign to your body, and then phagocytes can come along and basically gobble it up and clear it away. There are some autoimmune diseases like type 1 diabetes or Crohn's disease or MS, something like that, where things that are supposed to be in your body get opsonized. So when the beta cells in your pancreas are opsonized, you get type 1 diabetes. When myelin structures in your nerves are opsonized, you get multiple sclerosis. When components of the digestive tract mucosa are opsonized, then you get Crohn's disease. Okay, so that's when some of these things go wrong. But opsonization is just a general term. In fact, it just means enhanced phagocytosis. It doesn't have to be because of antibodies. I'm talking about here in the context of antibodies, but there are other chemicals that will opsonize pathogens to mark them for phagocytosis, besides just antibodies, but this is one of them. So I added opsonization on to the end of plan because while it's sort of what happens with all of the other four, sometimes it can be all that happens to get rid of it. Yeah.
Just wanted to ask a question about just the concept of solubility. So within a given solution, the solute eventually reaches the saturation point that eventually go beyond that falls out of solution. So does that imply with antigens that you reach a saturation point with a given number of antibodies?
No, it's not really treated so much in a chemical way. I did describe it showing you the silver nitrate reaction, but really when you're talking about bodily fluids, especially plasma, calling them a solution isn't entirely correct. They're really a suspension. And so blood cells are suspended in the plasma, and that's blood. They're sort of at the limit of size where they can stay suspended. But if you start agglutinating cells, or if you agglutinate smaller antigens and they make a large protein structure, then they can fall out of solution in a sense. But it's not quite the same as talking about it in the more strict chemical definition of solutions. So it's sort of appropriating the terms a little bit to describe biological processes. So, when you're talking about dissolving in solutions chemically in a biological system, you're really talking about hydrophobicity versus hydrophilicity. And it's not doing that. It's not changing the ability of that to happen.
Just the analogy made me question the mechanism a little bit.
That's fair, yeah. So, and if it's happening outside of the plasma in interstitial fluid or something like that, it's still a suspension essentially there, but smaller things can fall out of suspension. You get stuck into extracellular matrix structures or something like that. So this is not stated explicitly this way in the book. I'm really more, where'd it go? Sort of expanding on what's said down here in the function part. So we have opsonization, neutralization, fixing complement means lysis. I thought it said agglutination in here, but I guess I just said it myself. But IgMs are important for agglutination. So I also wanted to go back to another thing here on the B cell part of the book, which is, I was showing you this picture, and I was talking about primary and secondary responses. And this figure shows that a little bit more directly as what's happening in terms of being exposed to an infectious agent or an antigen of some sort. So on the y-axis, we have concentration of antibody seen in the plasma. And on the x-axis is time. Neither axes are labeled, but actually the labels kind of do play an important role. So from the initial exposure of something, so you get chickenpox for the first time or something like that, you're going to see an increase in the immune response in the number of antibodies that are produced and released from plasma cells. Now, this is going to take probably a couple of weeks to happen because the cells have to be exposed to the antigen to begin with, which does not necessarily happen instantaneously. And then they start to form clones, and then they differentiate into plasma cells and start releasing the antibodies. So it takes a little while to get to the point where lots of antibodies are being released. But eventually there will be enough antibodies, and it'll help your immune system fight off the infection. And then the antibody level goes down once the infection has gone away. And then sometime in the future, it doesn't have to be right at the end of the primary response there. This could be months, days, weeks, years, whatever in the future. You're exposed to the same thing again. And then with the next exposure, the concentration of antibody in the blood goes up very rapidly and very high. So if we just compare the peak of the primary response to what's going on in the secondary response, you hit that same concentration of antibodies pretty quickly, and you keep making a lot more antibodies. So instead of the couple of weeks worth of time that it takes for the primary response to kick in and fight off the infection, the secondary response is already to that level within days. And you're probably not going to really see symptoms of whatever's infecting you. So again, if this is chickenpox, the first time you're exposed, you have the primary response. And the next time you're exposed to chickenpox, the secondary response basically gets rid of it right away or within days. And you're not going to really be symptomatic of it. Okay. Now, the concentration scale over here on the side, the y-axis, doesn't have any tick marks. And it's unfortunate that it doesn't, because the scale here is actually a little bit unusual and not what you would necessarily expect. It's a logarithmic scale, which means that the difference between the primary immune response and the secondary immune response is not a linear difference. If we had tick marks here, it wouldn't be 1, 2, 3, 4, 5 times more. It would be 1, 10, 20, nope, sorry, did that wrong. 1, 10, 100, 1,000, 10,000. So the secondary immune response is orders of magnitude greater. And it's kind of unfortunate. A lot of books will leave that aspect out of it, that the secondary response is not only quicker, but it's immensely more robust response. And so once you've developed an immunity and you have memory cells ready to churn out more antibodies or memory T cells to do their job, you're going to get over the infection a lot quicker and probably before you're going to have symptoms show up. So like with chickenpox, you have the chickenpox once and then you're never going to have it again because all subsequent exposures are going to elicit the secondary immune response, this exponentially greater response that we see there. Okay? And it's because of those memory cells that can jump right in and do their job right away without having to wait for all of this activation stuff to take place. Okay? And in the book and in most books, they talk about this in reference to B cells and antibody concentration, but the same thing's happening for T cells also. They're not secreting antibodies because they're not B cells, but there are memory T cells, memory helper T cells, and memory cytotoxic T cells that are ready to go out and fight off an infection right when it starts off. And you don't have to wait around for a little while to get sick before it does its job. So, any questions about that? Can you think of an example where we can take advantage of that function in our body? Cold. Actually, the common cold is probably the worst example. What I'm getting at is vaccinations, and we don't have a vaccine against common cold. So if you expose somebody to a non-virulent form of a pathogen, say the virus without the genetic material present, it'll form the primary immune response, but there's no symptoms to fight off, so the person doesn't get sick. But they're ready for a secondary response when they ever are exposed to it again. Okay. The problem with the cold thing is that the viruses that cause the common cold mutate quite a bit. So when you develop a primary response to the cold one season, the next season you get exposed to it, you're exposed to a different pathogen, you're not immune to it. So that actually is sort of kind of the exact opposite of where you're going with. But if you vaccinate against something like the chickenpox virus or the polio virus or the measles virus or mumps or anything like that, you're basically making it so the body is ready to jump right into the secondary response when the body's ever first actually exposed to the virulent form of that pathogen. The next part of the book gets to talk about active versus passive immunity. But actually in active and passive, there's also natural versus artificial. Now, active and passive, as far as antibodies or T cells as involved, active is when it's your body making the antibodies. Passive is when they come from somewhere else. And then the natural form of that natural active immunity is when your system does this, when you get sick and then you're immune to it afterwards. Passive natural immunity is when you're getting antibodies from the mother through the placenta or through breastfeeding. And then the artificial version of active is when you're exposed to a vaccine, so you develop an immunity to it artificially, and then you're immune and have a secondary response later on. Artificial passive immunity is when you get immunity through some sort of therapeutic treatment. So this says immune globulin injections, which is fairly general. A good example of that is a snake bite kit. If you get bitten by a snake, have I said this before? Yeah, because I was talking about this back before we did the podcast thing. But just to finish that story, if you get bitten by a snake, the toxin in your system is going to kill you before you can go through a primary immune response. Okay, it's going to take you a couple of weeks to develop an immunity. You don't have a couple of weeks after a snake bite. So you inject antibodies against snake venom toxin from the snake bite kit, and you have a artificially induced passive immunity. Very rare situation, but that's a good example of it. Okay. So everybody has a natural passive immunity when they're first born. Everybody has a natural active immunity when they become immune to things on their own. And then it's fairly common that people will have artificial active immunity, and really people that don't are stupid. So because really everybody should be vaccinated against the things that we know we can provide that with. And if you don't, then you get the things like we see happening recently in Southern California with the measles or whatever that cropped up at Disney World, Disneyland, that sort of thing. People aren't vaccinating their kids, and so these things that shouldn't exist anymore because everybody's immune to them crop up again. And it's really because people aren't taking advantage of the technology we have for artificial active immunity. Yeah.
I saw this random thing about parents who have their kids vaccinated not wanting to have their kids interact with unvaccinated kids. But if your kids are vaccinated, what harm does an unvaccinated kid have to you?
That's a good question. I'm not entirely clear. I would think of it more as being, you stupid people, I'm not going to let my kids play with your kids. But I suppose there's a possibility that vaccines are not necessarily 100% effective, and so it's a little bit of an overreaction, I suppose, if that's the case. I think it's more, you're stupid kind of reaction. But I suppose it's possible that. I know another reason why it might be. There's some vaccines that aren't given very early because the kid's immune system can't handle it, or they're given in booster shots over a series of years. So if parents that are vaccinating their kids are going through this process, but they're not fully immunized, then if they're going to be playing with unvaccinated kids, then they might be exposed to something that they're not immune to yet, but they're in the process of getting immune to. Yeah, sure, we'll go with that story. Yes. I showed you this picture here earlier, which is really about helper T cells helping B cells become activated, which was left out of this picture here, but it does play a part in it. And that's the end of that section. The next three sections go into some particulars about immunology and certain disease states. And I don't want to say ignore those things. It's important that you look at them. But for the questions that I'm going to concentrate on, really meaning part 3 and part 4 assignments, I'm mostly interested in you being familiar with the the lymphatic system, innate immunity, and adaptive immunity, concentrating on the T cells and the B cells. Okay. Now, I do want to come back here at the end real quick to tie things back a little bit to what we learned in the podcast. At the end of this section about T cells, it talks about different types of T cells. And I showed you this picture, which is just T cells and helper T cells and cytotoxic T cells. But there's actually more. So there's a helper T cell, cytotoxic T cell, and there's also something called a regulatory T cell. Regulatory T cells are the lullaby cells that were mentioned in the podcast. They're also sometimes referred to as suppressor T cells because you could say they regulate the immune system, or more in the sense of a lullaby cell, they suppress the other T cells from going out and attacking things. So from the hypotheses put forth in the Jasper Lawrence story, the idea is cytotoxic T cells operating in a body that's been overly hygienized, if that's a word, don't have anything to fight off, and they feel kind of bored. So they're going to start attacking things that aren't really pathogenic, and you're going to get allergies or autoimmune disorders. That's the suggestion for what Jasper's story is talking about. And the suggestion is that having a low-grade infection of something like a parasitic worm is actually going to increase the activity of suppressor T cells, possibly to suppress overreaction to the parasitic worm, but then also that'll help suppress the attack cells from getting carried away and causing those sorts of diseases. That's really what that part of the podcast was getting at, how these things fit in with the other stuff. Okay. But as they said in the podcast, when you learn about the immune system, you learn about attack cells. That's what we've been doing. We've basically been talking about the attack cells, which primarily probably means the cytotoxic T cells, but to some extent the helper T cells help that do its job, and phagocytes and natural killer cells are also attacking things. I don't know actually the relationship of the regulatory T cells to the non-specific natural killer cells and phagocytes, but it might play a role there too. So, So anyways, that kind of ties it back into the podcast there. So I don't think you're the last class that I've done this. I think I have to do it on Tuesday next week too. But I have done this for several classes now, so I've rehearsed this presentation quite a bit. And I'm always afraid that I'm not going to get to finish things in time, but I'm actually done. So I'll call this the end for today. And next week we'll come in and we'll be doing the respiratory system.